So once we have our plasmid inside of our tube, the next step is going to be to perform a restriction digest. And so what that means is we're going to essentially want to take our plasmid and we're going to want to make cuts within the plasmid. So here we have the pET41-a plasmid vector, and we see there's these two sites where we want to cut. And so the way we're going to do that is we're going to add in some restriction enzymes. And so what these restriction enzymes do is they come in and they recognize specific sequences within the DNA, and they're going to make cuts in order to have a region where we can now insert our own gene of interest that we want to express. So to perform the restriction digest, we're going to be adding in cut smart buffer, as well as some sterile water, and our restriction enzymes. So the restriction enzymes we are going to be using are known as the NotI and NcoI restriction enzymes, and these are going to cut at specific sites. So for example, the NotI restriction enzyme is going to recognize this specific sequence within the DNA and is going to make a cut along where this red line would be. And so restriction enzymes, I like to imagine them to be like enzymes with scissors, and so what they're going to do is they're going to be able to go along and cut at that specific site, so that we then end up with DNA that has been separated and cut like that. And so similarly for the NcoI restriction site, we're going to have the enzyme go in and also cut along a specific site so that we can end up with another breakage in the DNA that looks like this. And so whenever our DNA separates like this, we call these to be forming sticky ends.